Hello students, welcome to EPG Pathshala program. I am Dr. Shoaib Lukman from the Council of Scientific and Industrial Research, Central Institute of Medicinal and Aromatic Plants, Lucknow. Today I am going to talk about the types of lipid, next part, from the paper Lipid Metabolism. The objectives of the present module includes another important type of lipid which is called as phytosterols. In this module, we try to know about the phytosterols, the significance of the phytosterols, and how they perform their function in a system. So, among sterols, phytosterols also have an important place. It was in 1878 when Hesse isolated the first sterols from the Calabar beans, which is botanically known as Phytostigma veneninosum, and he coined the term phytosterine. Later, in 1897, Thomas proposed the term phytosterols. These phytosterols have 24 alkyl groups, and each of these alkyl groups is distinct and very much conserved. The phytosterols has been found more efficient as compared to that of cholesterol in terms of expanding the temperature range in which the membrane associated biological process takes place. And this is due to the fact that plants have to face the higher temperature disparity than animals. So, we are discussing the different types of lipids, and these types include the simple lipids, complex lipid, and derived lipids. The simple lipids, including the fats, the oils, and the waxes, have already been explained in the previous modules. In the complex lipids, which includes the phospholipids, glycolipids, and proteolipids, has been discussed herein. Phospholipids are phosphorus containing lipids, whereas glycolipids are lipids containing saccharides or the carbohydrates. When lipids are amalgamated with proteins, it forms a complex mixture of lipids which is called as proteolipids. So in this module we are discussing about different types of glycolipids, and it has been continued from the previous modules. So today, in the types of lipids, we will be discussing about the complex lipid, and in complex lipid, the subtype glycolipids, and in particular, phytosterol will be discussed. As its name indicates, the phytosterols are unique sterols present in plants, and hence they get its name. But their close counterparts or similar analogues are also present in other organisms. As we have discussed that phytosterols have been more competent than cholesterol in expanding the temperature dimension in which the membrane coupled biological action takes place, and this competency is because of the fact that plants have to counter high temperature disparity as compared to animals. Mostly, the phytosterols have 28 to 30 carbon units with 1 to 2 carbon-carbon double bonds. Usually, one in the nucleus of the sterol, and occasionally a second in the side chain of alkyl residues. In plants, the cycloartenol give rise to phytosterols, whereas in fungi, including the yeast and vertebrates, they derived from lanosterol through the cyclization of squalene molecule. The facts also specifies the occurrence of biosynthetic pathway for phytosterol in plant cells via lanosterol, and hence was chosen as the lanosterol pathway. In plant species, over and above 250 diverse types of phytosterols have been identified and reported. Some of the envoys are campesterol, beta-cytosterol, and stigmasterol. It has been found that campesterol is a precursor of anabolic steroid boldenone. Beta-cytosterol is another phytosterol which is being used for the steroid biosynthesis, and it is present in all plant lipid. Similarly, stigmasterol, which is also known as Wilson factor, is needed for the synthesis of vitamin D3 and progesterone. Stigmasterol is a potential anti-inflammatory agent that inhibits numerous matrix degradation and pro-inflammatory mediators implicated in the cartilage degradation induced by osteoarthritis. In brown algae, the prevailing sterol is the fucosterol, and the chlorosterol exists in diminutive amount. Ergosterol, or else known as the mycosterol, is the C28 imperative sterol reported from the ergot and the yeast. The ergosterol was discovered by Tanret, followed by Gerard, who noticed this ergosterol in fungi as well. In yeast, Gerard was the first to identify ergosterol as a cell membrane component that is limited to fungi, and this molecule could be applicable as an indicator molecule for these microorganisms in an existing biomass. Upon irradiation, the ergosterol gives rise to calciferol, that is vitamin D2. Androsterol, which is also known as Argosta 7922 triene 3-beta-ol. It is an isomeric form of ergosterol and has been revealed from Antrodia camphorate, which is a fungus. This type of sterol has been found efficient in hepatoprotection and exert their effect through its anti-inflammatory ability. This compound androsterol is also used in Chinese traditional medicine for the treatment of cancer, diarrhea, drug intoxication, and hypertension. EuroFIR basis is one of the exclusive online database that contains the information on the content of the selected phytosterols which are present in different types of plants as well as plant-based foods. Phytosterols, which are detailed for an extensive portion of the total dietary sterols, especially in vertebrates, and they generate a broad range of biological actions in animals as well as in humans. They possess efficient cholesterol lowering and anti-tumor properties and decreases serum beta carotene concentration without varying vitamin A, D, and vitamin E absorption. In brown algae, for example, Sargassum ringoldianum and Lessonia nigricans, saringosterol, which is a derivative of phycosterol, has been revealed. And this saringosterol has been found to restrain the Mycobacterium tuberculosis growth. The effect was found to be comparable with that of rifampicin, but without considerate toxicity against the mammalian cell. In 2004, the European Commission endorsed the totting up of phytosterols as well as phytostanols in the food products with the condition of appropriate cataloging. Phytosterols, similar to that of cholesterol, undergo the oxidative processes and the resultant product oxyphytosterols have been revealed, and these oxysterols display some beneficial biological properties. Phytostanols are another group of, or rather subgroup of, phytosterols, fully saturated but with no double bonds, and are produced by the hydrogenation process. They are found in a very minute amount in the legumes, nuts, and the seeds, but the high concentration of phytostanol is present in few species of cereals. To augment the solubility, phytostanols combine with the esters of fatty acid to generate esters of plant stanols. Cytostanol is the utmost commonly found stanols that frequently crop up in dinoflagellates, and these cytostanols are the major source of 5-alpha hydrostanols in marine sediments. The 5-beta hydroxystanol and coprostanol constitute more or less 60% of the total sterols in human feces. In animals, these type of fully saturated sterols of the bacterial origin are conjointly established. Avenosterol is another subtype of sterol which is sequestered from the oil of oat and has been revealed to preserve explicitly frying oils from oxidation owing to its ethylidene group in the side chain. The phytosterols are of wide occurrence, and its persistence in the sediment has been reported in the geological samples of different age. The analysis, the diversity, the occurrence, and the health promoting uses of phytostanols and phytosterols have already been revealed and reviewed. Stigmastanols, dihydrocholesterol, and coprostanols are recurrently exist in sediments and occasionally utilized as the biomarkers primarily for the existence of life in petroleum and sedimentary organic matter. The majority of steroids exist in sediments are C27 and C29 steranes, which are pivotal intermediates in the exchange of steroids into steranes, that is, the oxygenated form to a saturated form. In addition, a wide variety of sterenes, which are monounsaturated, have been detected in the immature sediments. The marine desmosponges created 24 isopropyl cholestane, which is a hydrocarbon remnants of C30 sterol, accounted the charisma of metazone presence in the geological documents during the Neoproterozoic era. This sterol symbolizes a powerful biomarker for the early animal diversification and endows with the oldest or the primeval fact for animals in the fossil record. Sterol esters are the sterols that are often endowed esterified to the fatty acid. In the tissues of animals like adrenal glands, liver, plasma, cholesterol is esterified by an array of fatty acids and recurrently by essential fatty acids, accordingly forming the esters of cholesterol. The esters of sterols are indispensable and vital but exceedingly erratic part of yeast cell, with values varies from remnant to 50% of the total lipids. In animals, the cholesterol esterification inside the intestinal cells. For example, acyl-CoA cholesterol acyltransferase enzyme allocates the free cholesterol to be accumulated as a neutral lipid in the cytosolic droplets and in the stuffing of cholesterol into lipoprotein particles for the export by means of the liver cells via plasma. The human meibomian gland secretions like sebaceous gland at the edge of eyelids are predominantly rich in esters of cholesterol, exemplified by a monounsaturated or a saturated fatty acid moiety with carbon chain of 18 to 34. Cholesterol arachidinoate and cholesterol linoleate present in the nasal fluid have been revealed to bequeath to the intrinsic antibacterial activity. Cholesterol nitrolinoleate, a nitrate lipid perceived in the human lipoproteins and blood plasma, is a prospective marker of the chain-breaking antioxidant role of nitric oxide radical during the lipid peroxidation. Nitrated linoleic acid, which is a potent signaling molecule and a downregulator of inflammatory response, revealed to be fundamentally enhanced after macrophage activation, signifying that nitration of lipid happens as a part of the retort to inflammatory stimuli. Beta-cytosterol esters, stigmasterol esters, and ergosterol esters are present in plant cell membranes and the oils of the seed. Cycloartenol and esters of stigmasterol have been isolated from the bryophytes. In addition, cholesterol also forms ester linkage with secreted polypeptide and signaling molecules, preset by the hedgehog gene family during the growth and development of metazoa. The details about sterol esters, their synthesis, storage, and degradation have been well studied and reviewed. Another class of the glycolipids are the glycerophosphoplasmalocytosine. This trikentrione loaf is a Senegalese sponge, and from that sponge, an exceptional ether lipid has been revealed, which is distinguished by the glycosylation of two glycerol radicals and an alkenyl chain of ether, which consists of 24 carbon units with single double bond present at the third radical. In human brain, plasmalocytosine, a plasmal conjugate of cytosine and glycerol, which is an additional ether lipid, has been detected. Cyanobacterium, Cyanocystis species possess acylated forms of glycoglycerolipids bearing a palmitoyl group which is esterified at the hydroxyl group of carbon 6 of the terminal glycosyl moiety of either acylated DGMG or acylated